Hi students, it's Professor Bridget and welcome back to the cellular respiration lecture. This is part 2. So in part 1 we learned about the basics of cellular respiration and how in cellular respiration glucose, or the potential energy in glucose, is converted to ATP, adenosine triphosphate. Remember, that's the high energy molecule that cells can use to do their to carry out all of their daily chemical reactions. We know that cellular respiration occurs in the mitochondria, this organelle inside of the cell, and we also learned that the mitochondria is a double membraned organelle, and remember that membrane is made up of an outer membrane of a phospholipid bilayer, and then the inner membrane is also made of a phospholipid bilayer. And it's important that we recognize the importance of these membranes because the membranes are going to represent a barrier through which we can set up a concentration gradient. We learned about membranes and their importance in setting up concentration gradients. So remember, we're still kind of building on that knowledge that we've gained in the first few weeks of class. So what we're going to do now is we're going to go through the stages of cellular respiration. If you go to our Canvas website, you do have access to the PowerPoint versions of these lectures. And in the PowerPoint versions, I've embedded links that allow you to watch further explanations. So while watching this video, obviously you can't click on the links, but if you go to the modules and you go to the folder that has all of my PowerPoints in it, you'll be able to click right on these. I believe both of them are links to Amoeba Sisters videos, and they do a pretty good job of showing you in animation form the stages of cellular respiration. So let's just look at the names of the stages of cellular respiration. Remember that cellular respiration occurs in steps. So the first step is what we call glycolysis, and that literally means sugar breaking. So initially what has to happen is that glucose molecule is broken into two separate molecules. And the name of those two separate molecules are pyruvate. They're both 3-carbon molecules. Remember, glycolysis is a 6-carbon molecule. So if we split glucose right down the middle, we get 2 3-carbon molecules. So the second stage is called pyruvate oxidation. So you know that there's an oxidation reaction occurring. And then there's what we call the citric acid cycle. And then in stage 3, there is a process called oxidative phosphorylation. Phosphorylation, remember, is that reaction where we have ADP, adenosine diphosphate, and we add a phosphate to it to make ATP. So that's what phosphorylation is. When we add a phosphate to ADP, that's phosphorylation. So oxidative, again, tells you that an oxidation reaction is occurring. There are 2 substages to oxidative phosphorylation. There's the electron transport chain and what we call chemiosmosis. So we're going to go through each stage and understand how ATP is created from glucose. So when you look at this image from your textbook, it may seem a little intimidating at first, but what I'm going to do is we'll just go over just the basics right here first, so you kind of understand where each one of these processes occur inside of the cell. So this jelly bean shaped structure, that's a mitochondrion, right? And then over here, the authors are showing you that's the cytoplasm. That's outside of the mitochondrion. So if we look at these stages, the first stage, glycolysis, splitting glucose, occurs in the cytoplasm. And the two other stages of cellular respiration, the citric acid cycle and oxidative phosphorylation, both of those occur inside the mitochondrion. And one of the things that you see here is the representation of ATP being created during each step. The size of these little starbursts that you see here represents the amount of ATP produced in each step. So there's a little bit of ATP that is generated through glycolysis. There's just a little bit of ATP created through the citric acid cycle. The majority of the ATP is produced in that process called oxidative phosphorylation. And remember that we learned in the first lecture that electrons represent energy and that electrons are released when bonds are broken between carbon and hydrogen. And we learned about this enzyme. When it's not carrying electrons, it's called NAD. And so remember we referred to those as being electron carriers. So you see that in glycolysis, you've got NAD carrying a hydrogen and 2 electrons. So this being an electron carrier, NADH is going to carry those electrons, or essentially carry that energy into this final step where most of that energy is created. And you also see some NADH being created through the citric acid cycle. And you also see another enzyme here called, it would be without the electrons and the hydrogen, called FAD, but when it's carrying those electrons, it's called FADH, and that should be a 2 right there, FADH2, meaning that it's carrying 2 hydrogens and 2 electrons. So these are electron carriers also that, again, are carrying electrons and therefore energy to this last step, oxidative phosphorylation. So all of that energy being carried by these electron carriers is going into ultimately that third step. And that's why we get the most amount of ATP being created through oxidative phosphorylation in that very last step. So let's look at glycolysis first. Again, we've lost a little bit of formatting, and that's something that's kind of out of my control. But let's see the stages, or the chemical reaction that is glycolysis. So we start with a molecule of glucose. Remember, that's a 6-carbon molecule. 1, 2, 3, 4, 5, and 6. Remember, there are hydrogens attached to these carbons. Remember, the chemical formula for glucose is C6H12O6. So the authors here aren't writing in all of the oxygen or the hydrogens, they're just showing you the carbons, but assume that the hydrogens and the oxygens are there. So what's going to happen in glycolysis is that we're going to break this glucose molecule in half. That's what glycolysis means, right? Glucose splitting. So the first thing that's going to happen is, through this chemical reaction, we are going to split glucose into 2 separate 3-carbon molecules. NAD is going to play a role. Remember, NAD is that electron carrier, so there are 2 NAD molecules waiting to accept the hydrogens that are released from splitting glucose. And what you see happening to NAD is NAD picks up 2 electrons, and it picks up 1 hydrogen, and there are going to be 2 free hydrogens in the cytoplasm. Because we started with 2 NAD, we're going to end up with 2 NADH molecules plus 2 free hydrogens. And there's a little bit of energy that's created during this process, and that energy is used to phosphorylate ADP. So in the cytoplasm, there are going to be 2 molecules of ADP present and 2 free phosphates. So the energy that's released from splitting this glucose molecule allows for those two free phosphates to be added to ADP to make two molecules of ATP. So we've got a little bit of energy that's created in through this process of glycolysis, and then the products here are two molecules of something called pyruvate. So those are the products. And that pyruvate is going to be sent into the second stage of cellular respiration called the citric acid cycle. So let's have a look at that. So here we've got in the cytoplasm, glycolysis occurs in the cytoplasm. They're showing you here that we started out with glucose as a reactant, and through the process of glycolysis, pyruvate was formed. The electrons that are being carried by NADH, remember, they're going to be transported to the third stage, right? They're not immediately used in glycolysis. Here's a representation of the two ATP that are produced. And then also you see the pyruvate right here. That pyruvate gets sent into the mitochondrion. So now you have the products of glycolysis being sent into the mitochondrion, and they're going to be used in the citric acid cycle. So we will move now to the citric acid cycle, and a step that occurs just prior to citric acid cycle beginning. Before the citric acid cycle can proceed, pyruvate has to be modified a little bit. So here's pyruvate, that 3-carbon molecule. It's going to have one of its carbon bonds broken. And when that happens, again, remember you release energy, and the hydrogens are going to be picked up by NAD. And so, actually there's just, 2 hydrogens. And then, so that NAD is going to carry those electrons. So you see the 2 electrons here in blue. And that NAD will also carry 1 hydrogen, and there will be 1 free hydrogen minus its electron. The what comes out of that is 1 molecule of carbon dioxide, and then also there's an enzyme here that helps this process occur. We learned a lot about enzymes, and that enzyme is coenzyme A. So coenzyme A goes into this reaction to help it occur. So we start out with pyruvate. We've got some electrons being released and carried by NADH, and that pyruvate then gets converted into what we call acetyl coenzyme A. So check it out. We had 3 carbons, a 3-carbon molecule here in pyruvate, and that gets converted to a 2-carbon molecule called acetyl coenzyme A. It's acetyl coenzyme A that enters the citric acid cycle. Pyruvate does not enter the citric acid cycle directly. So remember that the citric acid cycle occurs in the mitochondria, and during each turn, 1 molecule of acetyl coenzyme A goes into the citric acid cycle, and the products after the citric acid cycle is completed, you've got 2 molecules of CO2, 1 molecule of ATP, 3 NADH, and 1 molecule of FADH2. This may sound a little intimidating at first, but these slides are really good for understanding what goes in and what comes out. And that's really what I would like you to know. So let's see how this occurs. Don't be intimidated by this graphic here. I'm not going to ask you to know all of the names of the intermediate molecules that are created during this process. what I'm really concerned with is that you follow the energy, follow the amount of ATP that's being produced, and follow NAD and NADH, because that's really what we're after. We're after where are the electrons going. We need to take account of where the electrons are going. But not only that, it's really important also to not forget about these hydrogens. These hydrogen ions are going to play a very important role in the next stage of cellular respiration, called oxidative phosphorylation. So let's go ahead and kind of follow each one of these little steps here. Okay, so let's look at what we start with. And remember, there are 2 molecules of pyruvate that get converted to 2 molecules of acetyl coenzyme A. This graphic is only showing you what's happening with 1 molecule of acetyl coenzyme A. You have to assume that this citric acid cycle goes through 2 turns, because 2 molecules of pyruvate get converted to 2 molecules of acetyl coenzyme A, but only 1 acetyl coenzyme A can go into the cycle at a time. So you'll have 2 turns of this ultimately. So remember, acetyl coenzyme A is a 2-carbon molecule, and it's going to enter that citric acid cycle. And when it does, the coenzyme A pops off. Remember, enzymes are recyclable. So coenzyme A really just acts as an escort for acetyl coenzyme A to make its way into the cycle. So coenzyme A pops off and gets recycled, and you can kind of assume that coenzyme A goes back to the beginning and waits for another molecule of pyruvate, right? So when that happens, when coenzyme A pops off, all you have left is this 2-carbon molecule. Look, because we started with that 2-carbon molecule. So the cycle is going to proceed, and that 2-carbon molecule, let's just say it's represented by these 2 carbons, is added to an already existing molecule in the cycle, but you don't need to know the name of that, okay? So 2 carbons get added to a 4-carbon compound to make a 6-carbon compound called citrate. And so citrate is the name of this compound, and really the reason why this cycle gets its name, the citric acid cycle. So now we have this 6-carbon compound, and as the cycle proceeds in this direction, we're going to break bonds again and release more energy. So here is NAD. When those bonds are broken of that 6-carbon compound, NAD is going to grab the 2 electrons and grab one of the hydrogens, and we'll also then have a free hydrogen. So we've got energy being carried by NADH, right? Another byproduct is carbon dioxide. So carbon dioxide leaves the cycle. And then if we proceed from 2 to 3, we started here with a 6-carbon compound, but we broke a bond and released energy. Now we have a 5-carbon compound. And the same thing is going to happen again. Look, we're going to break one of these bonds and release energy. That energy is going to be carried by the enzyme NAD. So NAD gets converted to NADH and a free hydrogen. And another CO2 leaves the cycle. Right? Remember, CO2 is one of the byproducts of cellular respiration. This is why. And at this point now, there's a little bit of energy produced to phosphorylate ADP. So here's ADP weighting, here's a phosphate weighting, and some of that energy from the 3rd step there is used to add that phosphate to ADP to create ATP. So in this last step, we had a 5-carbon compound, but we broke a bond, released a carbon, so now we're going to have a 4-carbon compound. And that 4-carbon compound then gets modified, but we're not we're not releasing a carbon here. That's not what's happening. Not like what we did in the first few stages. But some hydrogen bonds are broken. So now we've got this enzyme called FAD waiting to capture the hydrogen and the electrons associated with hydrogen. So FAD gets turned into FADH2. FAD can carry 2 hydrogens, so we don't have any free hydrogen here. So FAD is carrying 2 hydrogens, and it is carrying 2 electrons. So if we look up here, we still have a 4-carbon compound. So we haven't released a carbon, so therefore there is no CO2 released, right? Because CO2 would be formed by breaking a carbon off. But because we didn't break a bond between carbons, no CO2 is produced. So more hydrogen bonds are broken, and this NAD right here captures those electrons from hydrogen. It captures one of the hydrogens, and then we've got a free hydrogen. And so that's the last step in the citric acid cycle. And ultimately what happens is after step 5, you still have a 4-carbon compound left over. And that 4-carbon compound is going to be recycled right back into the citric acid cycle. And then it's going to be reused. So it will be this 4-carbon compound that gets added to that 2-carbon compound to create citrate again. So you see how this is a cycle. It just keeps spinning. As long as there is acetyl coenzyme A present, this citric acid cycle will continue to. So at the end, just kind of as a summary, at the end of the citric acid cycle, or the citric acid cycle processes 2 molecules of acetyl coenzyme A, right? So remember that pyruvate got converted to acetyl coenzyme A. But 2 molecules of pyruvate got converted to 2 acetyl coenzyme A. So for each for the citric acid cycle processes 2 molecules of acetyl coenzyme A for each molecule of glucose. So once the citric acid cycle is finished, it's going to go 2 times, right? So once it's turned 2 times, the overall products produced for 1 glucose molecule are 2 molecules of ATP, 6 NADH, and 2 FADH2. This energy right here, 2 molecules of ATP, isn't so much. We need a little bit of energy to keep this process going. Where the majority of energy is going to be produced is in the next stage, in oxidative phosphorylation. These electron carriers, and there are a lot of them, and they're carrying a lot of electrons and therefore a lot of energy, those electron carriers are going to carry that energy into the 3rd step. And so it's that 3rd step, oxidative phosphorylation, where we get the majority of ATP being produced. So let me just introduce the 3rd stage to you, and then we'll cover the specifics in part 3. So oxidative phosphorylation is the very last step in cellular respiration, and it's got 2 substeps to it. The electron transport chain and chemiosmosis. The electron transport chain is made up of a bunch of proteins that are embedded in that inner mitochondrial membrane. So remember when we learned about proteins being embedded in membranes? These proteins that make up the electron transport chain are found in the inner membrane of the mitochondria. So when those electrons make their way to that inner mitochondrial membrane, the electrons get passed off from one protein to the next, to the next, to the next, and there's a little bit of energy produced. So electrons from NADH and FADH, they're traveling down that transport chain ultimately to oxygen. Oxygen, remember, is going to be the terminal electron receptor. When those electrons are passed off from when they go through that electron transport chain, energy is released. The electrons are released. But we can't have free energy, right? Energy needs to be captured by oxygen. And remember, when the energy is captured by oxygen, so are the hydrogens. And so that oxygen ultimately gets turned into water. And we'll see that here in part 3. So that energy that is released as the electrons fall down that electron transport chain, the energy that is released is used to pump those free hydrogen ions from the what we call the the mitochondrial matrix into the intermembrane space. Remember earlier I said watch those hydrogen ions, they're going to be really important. This is where they come into play. So essentially that energy that's released is used to set up a concentration gradient where you're going to have greater amounts of hydrogen ions on one side of a membrane than you do on the other. And you know what happens with concentration gradients. Molecules want to move from high concentration to an area of low concentration. So we'll get to this in a second. I'm kind of getting ahead of myself. But ultimately, what's going to happen, as I said before, is oxygen is going to pick up hydrogen and create water. And then the last part is what we call chemiosmosis. And that's the process that actually phosphorylates ADP. Remember what phosphorylation is? Where you have a molecule of adenosine diphosphate and a free phosphate. So phosphorylation of ADP is where you add a phosphate to ADP to produce ATP. Okay, so let's look at a pictorial representation just of the electron transport chain. This isn't covering all of this third stage, but this is a good way to look at the electron transport chain. So remember that NADH and its electrons get transported to that third stage, right? And NADH drops off the electrons. So remember, it's kind of like this relay race, a passing of the baton. We're passing the electrons from NADH. So it's going to pass off its 2 electrons. And NADH is NAD is an enzyme. So look, it gets recycled, like all enzymes do, right? So we've got these 2 electrons now that get passed off to this protein in the membrane, and then that protein transfers the electrons to the next one and the next one. So these electrons are kind of falling down this staircase, being passed off from one protein to the next, and as that occurs, energy is being created. So you've got this nice, slow, controlled release of energy to make ATP, the synthesis of ATP. But energy doesn't go anywhere, right? We're not creating or destroying energy, we're just transforming it. So at the end or the bottom of that transport chain, you've got 2 free electrons, and you can't have free electrons bouncing around in a cell. That can be really damaging for a cell. So waiting at the bottom of this staircase or at the end of the electron transport chain is oxygen. And what oxygen is going to do is it's going to grab onto those electrons. You also have hydrogens waiting. And so this oxygen here is going to grab those electrons and grab those 2 hydrogens to make water. So before we end here, I just want to explain what the authors are trying to show you here. 1/2 of O2. Remember that O2 is written like this, right? That's 2 oxygen atoms combined into an oxygen molecule. So 1/2 of O2 really would just be 1 oxygen. Okay, so we've got 1 oxygen atom there, and that's going to accept 2 electrons, 2 hydrogens, and ultimately form water. So if we do a little bit of accounting here, we've got 2 hydrogens right here and 1 oxygen. So those 2 hydrogens come from here, the oxygen comes from here, and we end up with a molecule of water. So earlier, I think in part 1, I mentioned that oxygen is known as the terminal electron receptor, meaning that ultimately oxygen accepts those electrons. And that's why oxygen is so important in this process. This is why we need to breathe in oxygen, because oxygen accepts those electrons at the end of the electron transport chain. Okay, coming up now is going to be part 3 of the cellular respiration lecture. And in part 3, we'll go over the specifics of oxidative phosphorylation, that very last stage in cellular respiration. Okay, see you soon.